So welcome. Monica is going to, who's coming in from Portugal, is going to talk about the role of cytoskeleton in development and disease. Thank you, and please share your screen.
Thank you very much. It's really a pleasure to be here, also because I'm being quarantined, as I had a meeting with the Minister of Science in Portugal that had COVID, so I'm forced at home. So it's always nice to visit the world and not be forced at home. I'll share my screen. But I'm supposedly negative, so that's a good thing. Can you see my screen?
Yes.
Yeah, that's great. Okay. So again, it's really a pleasure to be here today. And I guess you have the questions on Slack if you want to, or I guess you can also put them in the chat, yeah? Or you can tell me about them, Ross. So just. Okay, so before I start, I would just like to say something about the institute where I come from, where you see all these people waving at you. And one of the things that we do have is that we really, we are a very international institute here in Portugal, and we really welcome, everyone from everywhere in the world. And so we do have PhD programs and postdoctoral programs. So just have a look at our website. So it's called the Gulbenkian Institute, and we have calls throughout the year. So have a look if you are interested into them. I think we may have had already people from this program. Additionally, what we also have that can be interesting from people from different places. So we had actually a program that you see here, which was a program dedicated to African students, a PhD program, which was actually Portuguese-speaking African students, that now is finishing for lack of funding. Unfortunately, we'll try to see if we can get more funding for it, and which was a fantastic program. And then we also have, for people that are interested in science education, we also have a program called the Lab in a Box, which actually is a program that has a lot of experiments that you can do in schools at different ages. And it's a program that we are actually implementing in several African countries, but also in Portugal. We also have a different program that hopefully will have a site soon called Lab in a Suitcase, whereby we are also developing equipment, cheaper equipment, so that people could also, have cheaper equipment to do experiments, to take them to schools, etc. So different things, and hopefully we'll also have that site soon. So the idea here is we have, like, one of our mottos is science from all for all, and so that we take science to everyone and everyone participates in science. Okay, so we also have programs for citizen science, for example. So now I'll go into my science, but you are welcome also to ask me questions about our institute at the end if you want to, or just write me an email. So I'm very interested in how cells are organized and how actually the different structures within the cell are organized and maintained in space and time, and how they really specialize into many different structures. And one of the structures here, you can see a lymphocyte, and for me, one of the most beautiful structures in the cell is this one called the centrosome. They are very, very tiny. So if you pick your hair and then you can cut it like the cross section to 100 times smaller, and there you'd have a centrosome. And centrosomes are tiny, but they have very, very important functions. So a centrosome, as I have it here, is actually composed of two centrioles, which are these structures, which are like 400 nanometers, 250 nanometers, so very small, as I said, and they are made of microtubules. And they have these two complete different personalities in our cells. So they can form the centrosome, as I said, which nucleates microtubules, which are these filaments that are important for many different things, such as cell division, cell motility, polarity, and also signaling. Many molecules go into the centrosome. But the centriole can also tether to the membrane, where it nucleates the formation of cilia and flagella. And cilia and flagella are also really important. And You can see here the centrosome participating in cell division, potentially also in cell migration, but just, you know, defining the microtubule network within the cell, which is important for transport of organelles, for example. But cilia, they really are important for many different things. So, for example, you have cilia in your cells at this moment that sense light. You have, those are not motile. They are signaling cilia. These cilia in your trachea, for example, they are expelling particles so that you know, they go into the right place in your trachea. You have also, for example, women have these also ciliated cells. They're important to transport eggs in the oviducts. You also have cilia in many different cells in our body that are signaling entities. And so actually, if you have problems in cilia and centrosomes, you can have many different diseases. So for example, here, if you have mutations in genes that encode for molecules that are important for the centrosome, you can have something called the microcephaly, where the brain is smaller. And actually, there was also a relation between the Zika virus and centrosome changes. I'm always losing my cursor. And also cancer. So if there are changes in centrosomes, they are associated with cancer. And I'll talk about that later on. But also you have, regarding cilia, cilia are really involved in many things, and they're because many cells have cilia. So if you have problems in cilia, you can have retinal degeneration, so blindness. You can have obesity because cilia are important to sense whether you've eaten enough or not in your last meal. You can have cystic kidneys, as you see here, the kidneys with cysts. You can have problems in digit formation and skeletal malformations because of many different signaling molecules that depend on cilia. So they're really important for many things. And actually, some of these structures were discovered very long ago, so more than a century ago, by these very famous biologists like Van Beneden, Bovary, Fleming, Morgan, and Loeb. And actually, they already raised many of the questions that we are interested nowadays in pursuing. For example, you know, what are these structures important for? Are they involved in human disease? How are they inherited? But it was really only in the last maybe 20 years now, so that because of proteomics and RNAi screens, we started to actually know what are the molecules that compose these structures. And knowing the molecules, we could look for them in different genomes and actually understand that some of these structures are very ancient. They were likely to be there in the last common ancestor of eukaryotes. So this is 2 billion years old. But on the other hand, we also started to have the tools to manipulate these structures because we can remove them or have too many of them in the organism. And that actually allowed us to address many important questions, at least for me. And in my lab, we are very interested in understanding some of the core principles of centriole and cilia biogenesis and maintenance of these structures. So for example, how do cells control the number of these structures? How do they control the length of these structures? How do they control where they form, when they form, and where they form? But also, we are very interested in how cells break the rules. So how do they create diversity within an organism during development? So how different cells have different cilia types, for example, how is that generated? Also during evolution, different organisms, how do they assemble these structures? Do they assemble in the same way? Do they assemble similar structures? But also in disease, you know, are these structures altered in disease? And how do they contribute to disease? So in our lab, we are very interested in these 2 types of questions. And today I'll talk about diversity in general. So how diversity of these structures is generated, and in particular during development and disease. And one of the interesting questions for me is really that, so we are, not you, because you work with fruit flies or you are interested in fruit flies, otherwise you wouldn't be here. But for many people that work on cell biology, they are very much focused on cells in culture, and therefore they are very much focused on very similar things. However, when we look into an organism, we actually see very diverse structures. So how do you generate these diverse structures? In particular, the cilia types, yeah? And in particular, this is a very interesting question because actually, these structures, at least the centrioles that are needed to make the cilia, they are most of them born in the same way. So this is a cell that is cycling or dividing. And in the G1 phase of the cell cycle, you have one centrosome with two centrioles. And then in S phase, when the DNA replicates, you also have these tiny centrioles being formed that will elongate. And so that then you have two centrosomes that then separate and help to form the mitotic spindle, okay? So, but in the next cycle, you'll have the same structures being born, which can form a cilia or not form a cilia, okay? And there are many things, interesting things here, namely, you know, that you only form one and only one, that you get the same length, and there's a certain pattern of aging of these structures. But for this part of the talk, I just want you to retain that actually they are born in the same way, however, they form different structures. So how do they do that? And of course, to do this, we could select different organisms in the tree of life, but the fruit fly is an amazing one because the fruit fly has different ciliary types. Cilia are important for, you know, proprioception, for hearing, for olfaction, for fertility, because they are part of the sperm. So actually, within the same organism, we can focus on different cilia in different tissue types, and we can look at, you know, how are they assembled and how are these different functions created. And this is what we wanted to do. So we wanted actually to look in detail at this part, which is supposedly born in the same way, and ask, are they different for the different functions, or are they all the same? So in order to do this, we actually had to describe them very well. And one fantastic technique to describe them is actually electron microscopy. So we can pick different cilia types of the functions that I was telling you, important for hearing, important for olfaction, and for fertility. And we could look at their very tiny structures in the different tissues of the fly and see what do they look like, using electron microscopy, in particular electron tomography, which actually looks at different angles of these structures so that you can have, in the end, a 3D structure, as I was showing here, of a cross section of one of these cilia types. And this is the membrane around, as you see here. And we can actually then make schemes based on these cross sections and longitudinal sections on electron microscopy. So for example, here you can see an olfactory cilia, and then another one here. And then here you can see a sperm cilia. So this is actually not the final flagella, it's a cilia that is formed before meiotic division, okay? And so what we can see here, for example, is that this is the cilia protruding out. These structures, which is the centrioles, are very small. They're like between 100 and 200 nanometers. These structures are really big. They are like 1 micrometer. These guys, you have two of them in a longitudinal fashion. These guys are perpendicular to each other. So these guys have doublets of microtubules. These guys have triplets of microtubules. And I can continue on and on and on about all the different things that these different cilia have. So this was for us very interesting because they're really, really different, okay? So what we could see was we looked at different parts. So if we look, for example, at the centriole, which is like the foundation of the cilia, as I told you, you could see the different sizes. We could also see that at the base, really base of the centriole, when a centriole is born, it actually has this structure, which is called the cartwheel, which defines this 9-fold symmetry of the centriole. So it's really beautiful. It's encoded by at least one molecule that really defines the 9-fold symmetry called SAS6. We were very surprised to see that these other cilia actually do not have it. Also here, in the ciliary gate, it's a very cool part because this is where it's the cilia, the cell decides what goes inside the cilia for signaling. And so this is a very important region, and it's called transition zone. And as you can see here, when we look at the transition zone here, we could see that the attachments to the membrane actually look quite different depending on the ciliary type, again, using electron microscopy. So this was really striking for us. So what we next decided to ask was, because we have the power of the fruit fly, we know the components that make these structures, so we could actually start asking about what is happening here. And we actually had many different hypotheses. So what we see here is that, they are different. So the bases are, seem to be different, of course, on a fruit fly, not the human here. So it could be that, they have different components, or it could be that they have the same components, but they are differently regulated so that you form different structures, or of course it can be both. What we do know is that when we look at the ciliary genes, many of them, the ones that we know, they are conserved across 2 billion years of evolution, as I told you. So what is there in a fruit fly, a human or a green algae, many of the molecules are common. We also know that if you have a mutation in some of these molecules, you can affect many different tissues, which suggests that many of these components are present in different tissues, okay? So it's not like different tissues would have different components. But at the same time, we know that if it's not a null mutant, it's a hypomorphic, and this happens a lot in human disease. Some of these molecules are actually very big, so you can have tons of different mutations that lead to human disease. And these actually can generate different phenotypes in the human. Okay, so some of them have more blindness, some of them have more cystic kidneys, depending on the type of mutation on the same gene, which actually suggests that perhaps the same genes might be important. So you have a core group of components that is important, but they might be regulated in a different way. So when you mutate in a certain part of the molecule, one of the tissues is more affected than the other ones, which suggests that this different regulation of the same components could be important. So then to test this, because we know the components that assemble, those core components that are present in many different organisms, we actually started to look into those. So we look at many, many different components. So more than, I think, 15 different components. And we characterize them in the different cilia types in the body of the fruit fly. And because these structures are very small, we actually use a super resolution microscopy. And what you can see here is what we can get. So this was already a while ago. So normally you see the cilia, it has tubulin, and at the base of the cilia you have one of the molecules that is part of there. And you can actually, those are similar to the cilia that I shown you before. So you see the two centrioles in a longitudinal fashion, but you cannot resolve the barrel of the cilia. And with this structural illumination microscopy, we get a twofold resolution, and you can start seeing the barrels very well. Actually, nowadays it's quite interesting because these microscopes are very expensive, but nowadays there's a different technique which is very cool that we started to do in the lab called expansion microscopy, where you actually you can embed these structures in a gel, and then it's like a baby diaper where you put liquid and it just really absorbs the liquid and swellens. And therefore, the structure becomes much bigger, 4 times to 10 times bigger. So now, and then we can put them in a normal microscope. It doesn't have to be one of these fancy microscopes. And that's super, super cool because you can actually get an even better resolution than what we have here. So that's something very cool. I don't have an image today here, but it's something that is, I think, very cool because you don't need very fancy microscopes to do that also. And so by doing this, I'm just going to show you an example of one of the molecules that we looked into to map, how different is the localization of these molecules or presence and absence in different cilia types. So we looked into this molecule that I told you, which is common in different, so that centrioles should be born with it because they have a 9-fold symmetry. And it's the molecule that is important to make this cartwheel, okay? Which the cartwheel, we don't see it there, okay? So what we saw actually was looking at, when these cells are born, and we saw that the centrioles still have this SAS6 molecule. However, if we look later on when the cilia forms, what we see is that you have the barrel, as you can see here, but you don't see this other molecule that supposedly should be there if the cartwheel was there and was there when the centriole was formed. And you can see it here very nicely. You can see the barrel, again, the longitudinal centrioles, as I told you, in the spermatocyte. And you can see the very nice cartwheel there, okay, at SAS6, okay. So we saw many different patterns. I'm just showing you here an example of something that was there in a tissue but is lost and is still present on the other tissue, okay, and generates diverse structures. We also observed some molecules that were there present in the different tissues, but have slightly different functions. So when you mutate them, you get different phenotypes, even if it's a null mutant, okay? So they seem to probably be interacting with different molecules. So I'm going to summarize this part of the talk and then move to a different one so that I can cover different tissues and different levels of diversity. So in this story, what we did see, you know, looking at many different components, was that we think that there's this canonical cilium, which has these core components, but actually what happens is that this is modified depending on the tissues so that you get different ciliary types, so in diverse spaces. So these components can be differently expressed. And that's, I think, very cool from our perspective on human disease, because as I told you, some of the humans that have these diseases that affect cilia, the cilia, they can have these different mutations, different alleles that affect different tissues. And we think it's because, the different tissues, in these different tissues, these molecules can be regulated in different ways, and therefore different mutations can affect differently the different tissues. So I think it provides a good way of framing what happens in human disease. And this could also, of course, be similar to other diseases associated with other organelles that are equally diverse between different tissues. I think it also raises many interesting questions from the point of view of evolution. How did these different cilia evolve, so that they have different functions, and how did the regulation of these different molecules evolve, which is something that I think it's also very interesting. And from the point of view of evolution, it's something that we are very interested in the lab also. I'm just going to show you a slide to show you what the top type of things that we are interested. So what you see here is what I've just shown you, which is the within organism variation. So different ciliary types with different structures in the fruit fly. But actually, if you look at the eukaryotic tree of life, so different, from worms to Naegleria, to ciliates, to green algae. So you can actually also see many diverse structures. So you see a lot of interspecies variation, which I think it's also very cool in terms of thinking, how did these structures evolve? So in terms of diversity, there's also something that I find very interesting, and I'm going to change gears now, which is again, this idea that supposedly, most of our cells are born with the same centriole number. So I was talking before about structure. Now I'm going to talk about number. And this is 2 centrioles. That's how they are born. However, if you look at our body, actually, you have different cells with different numbers of centrioles. So if you look, for example, in humans, but also in flies, if you look at muscle, skeletal muscle, actually centrioles are inactivated and lost in skeletal muscle. And in eggs, they are also lost. So actually it's super cool, because if you think about the fertilization, so the centrioles have been lost in the egg, and it's actually the sperm that brings at the base of the flagella the first centriole that the zygote sees. So while our mitochondria come from our mothers, our centrioles come originally from our father, okay? So you have asymmetric inheritance of these structures. And this is actually an interesting, this regulation of number can be interesting from the point of view that, different tissues have different numbers, but it also raises the question of why would different tissues have different numbers? It could be that you don't need them in certain tissues because you don't want to have this astral array of microtubules, so you lose them. That could be, like in certain epithelial cells, you do lose centrosomes. And more recently, different things have also been discovered regarding the regulation of centriole number. And so what we know, and this is actually not true in the fruit fly, but it's true in invertebrates, is that, so in fruit flies, this control is not as stringent, is that you have, in normal cells, you have this centrosome number, which is 1 centrosome or 2, depending on the phase of the cell cycle, and cells can proliferate. But if you remove centrosomes from our cells, or if you have too many of them, which you can do by manipulating some of the molecules that I was telling you in the beginning, then actually these cells will stop in the cell cycle. And this stop in the cell cycle is something that is dependent on a tumor suppressor, p53, okay? This is actually very interesting because you could think that, you know, if cells regulate their centriole numbers, could be a way of ensuring that they are stopped in the cell cycle and they would not over-proliferate and generate cancer, for example. So it could be an additional control of, you know, proliferation. So, and it could be important, for example, for regeneration also. So already long ago, one of my students in Bento, she was really curious about, you know, how are centrioles regulated and how do they disappear in the eggs? And of course, we wanted to look in the fruit fly because it's so much easier to study the fruit fly. And she also had the question of, you know, what is the relevance? Why do eggs have 0 centrioles? What would happen if they don't lose the centrioles, okay? And this is a project whereby Anna Marques actually joined later on in Bento in the project, and she helped Inês to finish the project. And so at the time, we actually thought initially, okay, we are working fruit flies, let's do a screen whereby, you know, we check if we can find mutations where centrioles do not disappear and then identify the genes that are involved in regulation. Then we really did not have a good positive control because we don't know of anything that regulates this process that could tell us, you know, what would happen. So we decided just to do, you know, the classic biological thing of just observing and seeing if we come up with a good hypothesis. And so at the time, what Inês decided to do was to actually look at centrosomes during oogenesis in the fruit fly. And so she looked at the 2 components of centrosomes. So you have the centrioles, as I told you, and then you have the material around the centrioles, which is called pericentriolar material matrix, which is actually needed for the nucleation of the microtubules because you have molecules there that nucleate and also anchor the microtubules. So she just decided to go look during oogenesis and see what was happening. And so what you have in, I guess you probably already had people talking about oogenesis in the fruit fly, but you start with a stem cell that divides and you end up with a cyst of 16 cells, and only one of them will become the oocyte. And then these other cells called nurse cells actually dump a lot of material into the oocyte, in particular the centrioles. So actually the Drosophila oocyte is quite unusual in that sense because it really has, at a certain stage of its development, a lot of centrioles. So it should have around 64 centrioles from all the different cells that are deposited there. And then we knew that somewhere before meiotic division, the centrioles should be lost. And the question is, how are they lost? Okay, so she decided to look at all this process and look at the centrosome and the centriole and see what is happening there. So what you can see here is that you actually see, if you look at early, middle, and late oogenesis, and then in meiosis, you can see that the material that surrounds the centriole actually goes down with time. Okay, so you have much less pericentral material, and centrioles are actually quite stable. And it's only in meiosis that, you know, we're seeing the spindle, but we don't see centrioles around in the oocyte. And so we could actually quantify the number of centrioles with time. So it's an approximate centriole number because they form a clump. So we look at the intensity of the signal. And in mid stages, you see equivalent, you know, to a lot of centrioles. It could be almost close to the 64 centrioles that we would expect. Yeah, so it's not far from it. In stage 10 of oogenesis, you'd still have them. And it's really later on that they are reduced, and we don't see any of that in the division. So what we started to think, you know, looking at the pattern that I described before, which is that you lose first the pericentricular material, and which is the blue material, and then afterwards you lose the green material, the centrioles, that maybe this material that surrounds the centriole somehow protects them. And when you lose that, then you lose the centrioles. And this was just an hypothesis. I told you it was starting just with observation and seeing what happens. I'm not following the Slack channel, so if I should be following, let me know, okay?
Yeah,
So you
no
can
worries.
interrupt me. Okay. So what the hypothesis would be something like this. First you lose the blue material and then you would lose the centrioles. So to test this, we actually, first in tissue culture cells, we can do RNAi. So we can remove different components of the blue material by doing RNAi and ask whether in those cells in culture from the fruit fly, do we lose the centrioles or not? Okay, and this is just looking at centriole loss. This is a control. This is when we remove all these materials. So what we see here is the individual components of that material, and this is when we remove all the 4 components. And if we remove just individual components, the stability is similar to a control. This is just M cherry. The cells do not express M cherry, so this is just a negative control. It could be GFP RNA, double-strand RNA. And here we remove all the 4 different components. And what we can see is really an increase in cells without centrioles. And if we leave it even for longer, we have a bigger increase. So clearly, if we remove this matrix, we have the loss of centrioles, at least in tissue culture cells. Then we try to overexpress some of these components in the eggs to see, can we prevent the loss of these components and therefore maybe prevent the loss of centrioles. And single components were not sufficient. So we started to think, can we regulate these together? Like, is there any molecule that regulates all these components so that we could play with that molecule instead of playing with individual components? And so how is this matrix regulated? And we actually know that there's a kinase called polo kinase, which is very important for cell division and also for cell cycle progression, that in many different cell types, in different organisms, actually when a cell is in interphase, it has a small amount of this blue material, the matrix, and then polo kinase actually brings a lot of this material that is needed for enriching the centrosome in mitosis to help forming the mitotic spindle. So we started to think that maybe what was happening was that it was a reverse. Maybe the cells, the egg, is losing polo, and therefore the centrosome is shrinking, and this is what is happening here. And so we actually look at polo in the microtubule organizing center, so the centrosome of the oocyte, and we saw a very similar pattern as compared to this matrix material. We also looked at the stage where normally the centrioles are there in oocytes, and we did RNAi for polo at that stage, and we saw less centrioles, and also in tissue culture cells. So polo seems to be important for the stability of centrosomes. So now we could actually do, I think, one of the most interesting experiments, and the one that we wanted to do from the beginning, which is, can we actually prevent the loss of centrioles? Now that we know the molecules that play a role, can we actually put polo there and prevent the loss of polo, and therefore prevent the loss of this matrix, and therefore prevent the loss of centrioles, and therefore keep centrioles during the meiotic divisions, and ask what would happen to the oocyte? Okay, so we started by just overexpressing polo, and actually it does not go to the right place at the stage, at these later stages. So there's some reason why it actually is coming out of there. And if you just put polo, that's not sufficient to go there, okay? Probably because the molecules that tether polo are also going down in terms of transcription. And so we started to think, can we actually play a trick with these cells and keep polo there, okay? And in our field, there is a small domain of a molecule that actually tethers. It's a domain that localizes that molecule to the centrosome, and we can use it ectopically fused with the other molecules to tether them to the centrosome. So we actually fused polo to this PAC domain and asked, can we keep polo there, okay, and expressed it in oocytes. So what you see here is that we just expressed GFP fused to this domain that tethers molecules to the centriole. So GFP actually would go to a centriole if a centriole was here, but we are looking at female meiosis and there's no centriole normally, and that's what you would expect to see. However, when we tethered polo to pact, what we actually saw was quite nice, which is that the centrioles that normally should have been lost, they're actually here in the oocyte and really nucleating many, many microtubules, okay? So we saw that the centrioles were normally retained in all the different oocytes that we looked into, which was quite nice. So polo is sufficient to keep the centrioles. Of course, then we wanted to ask, so if we now keep the centrioles, what will happen? Yeah, of course, Drosophila, it's like the worst case that you can look into. I mean, the worst in the sense that, it has so many centrioles that if you keep all of these, it would be unusual if they can really, survive and, be fertilized and have a normal embryo. And actually they don't. So meiosis already is abnormal, as you can see here. These asters also start, interacting with the DNA that you can see here in blue. But we can also see after fertilization, this is a normal syncytial embryo with the centrioles and the DNA and the spindle. But if you have polo tethered to the centriole, and actually you start, you have extra centrioles, and as you can see here, and they start interacting, through the microtubules with the DNA, and they really don't pass through the first division. So they just die. So these females are really sterile. So to summarize this, what we know from here is that normally in eggs, this matrix that surrounds the centrioles is somehow disappears, and that induces the centrioles to disappear, so that the egg does not have any centriole, and it's the sperm that contributes with the centriole that then is needed for egg development or zygote development. So I think here there's a cool thing, which is that this matrix that we thought its main function was microtubule nucleation. We have a different function, which is that it protects the centrioles, which is, we are trying to now understand how does it do it. So what is the mechanism of protection? Does it protect things to go inside and destroy the centriole? Does it protect or keeps things inside from going out? Many different hypotheses. And of course, we are very interested to know how universal and how relevant is this to other tissues. In particular, in the fruit fly, it's nice because we have many cells that have centrioles for a long time, like ciliated cells. And you have cells where the centrioles disappear, like muscle. And so it's cool to see whether you have deregulation of this matrix here, and whether you maintain the matrix here. Yeah? And we're also looking into regeneration in salamanders to see whether this regulation of centrosomes is important for regeneration, because centrosomes in vertebrate cells are needed for cell proliferation. So I would finally very quickly go into the last part of my talk, which is into human disease. And here I drift off from the fruit fly just for a bit, I'm sorry. So we do work with different model organisms in our lab, depending on the question that we have. We can work with fruit flies, with plant cells, with yeast, with mice now, with salamanders also in collaboration, and with human cells and cancer tissues, okay? And so one of the questions that I told you is this regulation of centriole number, which is actually quite strict in most of our cells. However, we actually know that in cancer, this can be deregulated. And the question is how, does this happen, and whether it is important for the development of cancer. This is actually a question that was raised already long ago, and Bovary and Van Beneden were people that raised this issue. And what you can see here in a slide from Bovary is that in an egg that was fertilized by two sperm cells, then this egg, the centrosomes also duplicate. So you end up with 4 centrosomes, and the DNA does not divide normally because, you have not the normal 2 poles, and you can generate an euploidy. So he already had, more than a century ago, the hypothesis that if you have deregulated centrosome number, that could generate an euploidy, and that could be involved in cancer. Okay, so for long ago. And meanwhile, then people started to actually look into different types of cancer. And they started to observe that actually you could see the regulated centrosome numbers in different tissues, in different tumors, okay? Not in normal tissues, but in cancerous tissues. But of course, this actually is something that, when you think about cancer, cancer cells are deregulated. And of course, the question that arises is what is the chicken and what is the egg? Are centrosomes deregulated in cancer? Because cancer cells are deregulated, and therefore if they are deregulated, maybe the components of centrosomes are deregulated and you have too many. Or could actually centrosomes play a role in the development of tumors, okay? Or maybe in some tissues is one way, in one type of cancer is one way, and the other type of cancer could be a different way, okay? And actually through, these molecules that I was discussing in the beginning that we now know are needed to form centrosomes, we can actually regulate the number of centrosomes. There's a molecule that we work with also called PLK4, which is called polo-like kinase 4. So it's from the family of the polo-kinase that I just told you about, but it's a different polo-like kinase. It's a bit more diverse. In flies, you only have two. It's polo and this polo-like kinase 4, also called SAC. And that, it's downregulation, you don't have centrosomes. Too much of this molecule, you have too many centrosomes. And actually the experiment was done by different labs, and I'm just going to show you one here, whereby done by Levine and Andrew Holland's lab in the States, where what Andrew saw was that if you this is a mouse, if you have normal levels of PLK4 in the control, you are looking at tumor-free survival and you don't have tumors. If you have too much PLK4, and they saw that it generates too many centrosomes and could generate things like this, you actually have lymphomas, sarcomas, and squamous cell carcinomas, and the mice die, okay? So centrosomes can actually promote and enhance cancer, okay? So this is something very interesting and something that we were also very curious to look into, okay? So we were curious to know what type of central abnormalities exist in cancer and how penetrant are they, and what are the causes and consequences of these abnormalities. And so in order to start to do this, we've done different approaches. What I'm going to show you today is one where we look at different types of tumors. So we look at the NCI 60 panel of cancer cell lines, where you have leukemia, you have breast, colon, lungs, different types of cancers. And it's a cool panel because you have the genome is characterized, gene expression, drug resistance. And this is something that we did together with David Kalman, Susana Godinho, now at the Barts Institute, and Joana Paredes, André at IPATIMUP, Nuno Moraes and Bernardo at IMM in Lisbon. And this was a project driven by a postdoc, Gael Martei, together with another postdoc, Adan Guerrero. Gael is back in France and Adan has his lab in Mexico.
Great, Monica, if you can do it quickly, that'd be awesome.
Yes, I'm really finishing. So we did this screen, and where we looked at the centrioles in the different cell types, we quantified these in an almost automatic way. And what you see here is some type of abnormalities that you see. You see here normal cells, okay, with the normal centrioles that you see at the poles and the DNA and the spindle. This is a normal cell in cancer. It also happens. And this one, it's another cell in cancer where you see many, many centrioles or really long centrioles, for example. And so I'm just showing you an example of quantifying this. If we look at the different cell lines and the percentage of cells that are deregulated, so with more centrioles, we of course had to look at normal tissue or non-cancerous, and you already see some changes. It's actually quite difficult to find human cells that are normal and can survive in culture. And we looked at the different tumors, and you see many of them deregulated. I'm just going to show you something that was very cool. So we observed actually that here, when we look at the breast cancer, we saw here cell lines that are of a tumor type, luminal, which is less invasive, whether these guys that are more deregulated are actually of a tumor type, breast tumor type, that is more invasive, which is a triple negative breast cancer. Of course, these are cell lines, they could be completely deregulated because they've evolved in culture. So we wanted to look into tissues. And so we actually look into tumors themselves. And so we look at luminal tumors and basal tumors, and then again, looking at deregulation, we see the basal ones are also more deregulated in terms of the central number as compared to luminal. So to summarize, what we saw was that these changes are actually a hallmark. It's very common that we see the regulated number and size of centrioles in different tumor types. It's associated with bad prognosis. In certain tumors, we actually characterize a long tumor progression in the tissue, like in tumor of esophagus, Barrett esophagus, and we saw that changes can occur even before the cancer arises. And so, of course, it raises a lot of interesting questions. So what are the molecular mechanisms, drug sensitivities, this association with prognostic, the evolution of these cells, what happens to the cells with multiple centrosomes, and of course, the validation in tumors. And I'll just go back to this to summarize the first part again. So what I told you is that there's also, besides this diversity in disease, there's also diversity in terms of our tissues, in terms of how the numbers are regulated. And I told you that actually some cells have 0 centrosomes, and that this material that surrounds centrosomes is very important to maintain these structures. And I think understanding how this is regulated can probably be quite important from the point of view of understanding how cells regulate their differentiation and when they stop cycling. But I've also shown you that in our cilia types, these bases can also be quite different, and that these core components can be differently regulated, which I think it's very interesting to understand also human disease. And I would like to thank the people in my lab. So this is what was just before the lockdown. Retreats like this would not occur now. Hopefully they will occur soon. The people that I've showed the work today. So Shadin Jana is the person doing most of the ciliary work, also with Sasha, who is not here in the picture. Anna Marques is one of the persons together with Inês Bento doing the oocyte work. And Gael Martey and Carla Lopes are doing the cancer work, and our collaborators and these institutions for funding. And thank you very much for listening. I'll stop sharing.
Yes. Excellent. Thank you, Monica. Really appreciate it. Really beautiful. Centrals, something I need to know more about. So we have a number of questions, so I'm just going to condense to a few, and then, as you say, you can move to Slack as you wish. Actually, you know what? There's a number of questions from Mohammed Jamai. Mohammed, you want to get online and you can ask them yourselves, because you've posted a few of them. So why don't you go ahead?
Yeah. So I thank you for the presentation. It was wonderful. So I'm just actually interested in So I have actually a central idea about the centrosome organization and the centrosome number in relation with migration and metastasis. And the main question of my actual work is, if we think, or if we can imagine that the overexpression of the centrosome number in a cluster way can reorganize, the cytoskeleton and the actin filament, and this could help cell to migrate more or to migrate in a different way, And if we could, by clustering them, changing some, overexpression, low expression, some protein, etc., if we could convince cells to decluster and then to reorganize the forces and to reduce the migration. So all this fantastic, I assume, way of thinking, if you have any idea about this in your experiment.
Yes, so actually there are some people that actually looked at the role of centrosomes when they are amplified in cell migration, because that's actually one of the cool things in terms of when you think about tumors and what may happen when you have too many centrosomes. And I didn't say, but the multiple centrosomes can actually, as you say, generate more microtubules, and that affects many things, in particular the capacity to metastasize, the capacity to secrete molecules that induce other cells to metastasize. So there's many different things that actually have a role there. But there is work done looking at what happens if you have multiple centrosomes. I think in endothelial cells, that's there's a paper in JCV already a while ago. I mean, if you send me an email, I can try to look for that paper. And I think in that paper, the idea was actually that the cell migration was not as organized as it would be normally, okay? And I think, was there another one recently? I can have a look at it, okay, looking into that. So, but definitely, so what you're saying is that if you have too many centrosomes, depending on how they are organized in the cell, of course, you have different networks of microtubules, and that could influence the way cells behave, and that's true. If the centrosomes are very clustered, which is whether they come together, which is something that very often occurs in cancer cells, as you probably know, this is a way of cancer cells adapting to having many centrosomes and not having, you know, everything going in every direction when they divide and then dying. So that's to cope with that, they cluster their centrosomes. And of course, clustering their centrosomes, which seems actually to be the case in many cells in interphase that have multiple centrosomes, could also have an impact in cell migration, as you are saying. We start to know some of the molecules that actually play a role in clustering. So that could be a way of playing with these, as you say, so that to prevent the clustering and force them to come apart. And some of these are some kinesins. If you send me an email, I could also send you some references.
Absolutely, absolutely. Thank you very much. Merci. Thank you.
Thanks, Mohamed. All right, and the last question here. So Kamaldeen, we're gonna answer your question, which is a technical question on Slack, because that's a complicated answer. So Hamid Badmos, actually, why don't you go ahead and ask the last question, Hamid, since you're one of the speakers here. Actually, last question, then we're gonna talk. Go ahead.
Right. Thank you very much, Ross. So my question is basically, so you're showing the kinases, the polar and polar-like kinase. So are there any phosphatase that can act in reverse to these kinases in your system?
Yes. So definitely. So there are different phosphatases that PP2 is one of them that actually counteract the phosphorylations by polar-like kinase 4 and regulate centriole number. So definitely, yes. So whenever there is a kinase, there's always some phosphatases that play a role. Yes.
Okay, thank you.
Thank you.
All right, Monica, I'm going to put the rest into Slack.
Sure. Thank you very much, everyone.
Thank you very much. A little applause would be good. Here we go.
And enjoy the classes.
Yeah, no, much appreciated. You taking some of your valuable time.